Okay, so we just talked about what the electron transport chain and chemiosmosis is. Now we are going to dive into the electron transport chain. But before I do that, I want to go over our formula for cellular respiration first, because I'm going to help you guys organize some stuff here. So we have glucose plus oxygen turns into CO2 and water. So we get 6 oxygens total, 6 CO2s, and 6 waters. Before we get going over here, I want to do an inventory of all of the molecules that we've made so far during cellular respiration. So if we remember in glycolysis, we start with a 6-sugar compound. We invest 2 ATP. So we're going to do this as a net gain in what we've got. And after glycolysis, we've harvested 2 NADHs. They had been reduced from the breakdown of sugar. We also ended up with 2 ATP. We also had 2 pyruvate. Those pyruvate go into the link reaction. And then from the link reaction, they go into the Krebs cycle. In the link reaction, we get our first CO2, and that's going to happen for each pyruvate. So you get 2 carbon dioxides. We also get 2 more NADHs. Pretty awesome. And then our little acetylcholine moves into that cycle, joins with oxacylate to make a 6-carbon sugar, and then we continue to decarboxylize and oxidize that compound. And in the Krebs cycle, we end up with a total of 2 more ATP. We get 6 NADHs and 2 FADHs. Okay, so remember, these are reduced from NAD to that by oxidizing all the molecules in the Krebs cycle. That gives us a total of 10 NADHs, 2 FADHs, and I forgot, we also get 4 CO2s here. Okay, 6 CO2 and 4 ATP. For net ATP, we actually have 6 total ATP. So far in our breakdown of sugar, is there really any of the carbon from sugar left by the time we finish the Krebs cycle? It's gone, right? It's now all 6 carbons have been removed into CO2. So really at this point, this is all the Krebs, link, and glycolysis. Have we used oxygen yet? Not a bit. Have we created our CO2? Yeah, that was all in the link and Krebs. Okay. And we have not produced any water yet. So this is going to be our electron transport chain. I'm going to draw real tiny here. I hope you guys can see this. It's hopefully better. We have our mitochondria. Okay, it's got this inner membrane, a bacterial inner membrane, and then a eukaryotic outer membrane. And we're going to really be focusing on this cristae right here. Okay, the inner membrane of the mitochondria and this space in between, the inner membrane space. So we're going to zoom in on that, and that is going to be what I'm going to draw up here. So I am drawing just a small little tiny section of this cristae. This would be going on along the whole cristae. Lots of invaginated folds because we're increasing surface area so that more and more electron transport chains can occur. So this is going to be the cristae, or the inner membrane. Okay. We're going to claim that this is the inner membrane space. Okay, so this would be inner membrane space. This area in here, this would be the outer membrane, and this here is going to be our matrix. Okay, the cytoplasm of the mitochondria. Okay, located all throughout the membrane are electron carrier proteins. You don't need to know the name of them, but some of them are called like cytochrome C. We talked about that one with in-product inhibition, because if it's inhibited, it'll actually shut down the electron transport chain, and that's what cyanide does to kill people. But all the way through here, we have integral membrane proteins. These are going to be channel proteins as well as electron carriers. And we typically say that there's about 3 of them. Really, they all just run in series, and there's millions of them, really. We'd add about 3 electron carrier proteins. And then the most important protein, and everybody's favorite is ATP synthase, which personally kind of looks like kind of circular, and it's got a little mechanism in here. This is supposed to represent spinning. I'm not the best drawer in the world. But this thing actually turns kind of like an engine on a jet plane, and it spins around like this, and this is ATP synthase. Okay, so ACE means it's an enzyme. It's an enzyme that's going to make ATP. These are the ETC proteins, or enzymes. Okay, electron carriers are what they are. So what's really happening here is, in the matrix, we have made all this NADH and FADH. All of these are carrying high energy electrons and hydrogens. So as an NADH comes up to this enzyme, it is going to be oxidized back to NAD. Okay. What happens here is it's going to drop off its electron. Hoping you guys can see this in red, and it's a really high energy electron. So that energy electron, lots and lots of energy. It takes special molecules like this enzyme to hold that energy because it's so strong. And it's going to use the energy from that electron to actually pump hydrogen into the inner membrane space. So we have now oxidized NADH, which was actually, the energy came from that, from oxidizing glucose. So this is why we call this oxidative phosphorylation, because we're oxidizing this molecule. We're oxidizing glucose to get the energy to create this proton pump, proton being hydrogen pump. So we're pumping protons up into the inner membrane space. The same thing is going to continue to happen. You may also have FADH, also become oxidized and release its hydrogen up to here. All the energy comes from this, from electrons slowly losing energy as they're passed along the electron chain. So the big problem with these NADHs, why we can't use them for most of our energy sources, is because there's too much energy. It would be so much, the cell would actually probably explode or have damage done to it. So these electrons have to slowly be passed down and their energy slowly harvested as they go down the electron transport chain. Once the electron gets here, the electron is spent. It doesn't have a ton of energy left, maybe just enough to pump some more hydrogen up here, but that electron is now spent. And what this whole process has done, and most importantly, has created a lot of hydrogens on this inner membrane space. So what we've created is a proton gradient in the inner membrane space, where we have a really high concentration of hydrogen here and a very low concentration of hydrogen here. And if you think back to diffusion, molecules want to move from areas of high to low. So without any energy, all the energy was put in here to pump these protons in. Without any energy, these hydrogens want to get back down. And they're going to be allowed to come back across the gradient through ATP synthase. And the power from the hydrogen coming through ATP synthase, the energy created by this proton gradient, is going to cause this ATP synthase to spin a little bit, and it is going to then phosphorylate an ADP to ATP. Make sure you guys can see that. Okay, good. So we have now phosphorylated it to ATP, and we're actually going to make about 30 of these. In a perfect situation, this would happen 30 times for every glucose. Life is not perfect. Some of these hydrogens don't work. Some of the electrons aren't as good as you think, or they end up somewhere else. So really, this is in a perfect world. We should make 30 ATP here, which gives us a total of, actually, I'm sorry, that should be 32 ATP. You actually get, for every NADH, you get 3 ATP, and for every FADH, you get 2. So it's actually 34 ATP. I'm sorry about that, guys. 34 ATP for a total of 38 ATP. 36 to 38, because we used 2. We've created this proton pump. It's come back down. Now, one thing you should be asking is if all the hydrogens are coming back down here, we're losing our proton pump. And a key IB question, and it's actually just, why do we need oxygen? And this is where we need oxygen. So all of this, we have now not used any oxygen. We have not created any water. We have all these hydrogens coming down. Okay, and we also have this spent electron. That electron needs to go somewhere. Oxygens are really negative. They like negative things like electrons, so they actually want 2 electrons. So oxygen is now going to be the final electron acceptor at the end of this. So oxygen, this is the whole reason we need oxygen. It's the whole reason we breathe. Why oxygen is so important is for this final step of cellular respiration and the final step of the electron transport chain. Oxygen is going to take that spent electron, and it's also going to join with 2 of the hydrogens that come out, and we produce H2O. So these hydrogens, to get rid and help keep the gradient, are going to attach 2 to every oxygen molecule, and oxygen is the final electron acceptor, and we now produce our water. So overall, for every glucose, we would end up producing 6 waters after we used 6 oxygens. That is it. That is the electron transport chain. That is how we make ATP inside the cell, or more importantly, inside the mitochondria. This same process is going to occur in photosynthesis. It's going to occur inside the chlorophyll, and we'll do that next week. Inside the chlorophyll, the only difference is, instead of getting energy for the electron transport chain from the oxidation of sugar, we're actually going to get the energy to pop these electrons out from splitting water and the energy from the sun. And you'll really see how the whole cycle goes back to each other. I hope that was better than me drawing on the computer. Make sure you try to draw this out yourself. Really understand that we create a hydrogen proton gradient in the intermembrane space, and that is used to power ATP synthase, and oxygen is the final electron acceptor. By the way, chemiosmosis we talked about before. This is chemiosmosis right here, where we have a proton gradient created by the oxidation of sugar, and that proton gradient is going to power ATP synthase by running across the gradient. Osmosis, right? Osmosis would be water. This is chemical osmosis in the form of protons. That is good. I'll go back on the computer in a minute, and we will go over how you would write up a long answer question for electron transport chain.